Hello and welcome to Effortless Mastery. Today we'll be learning about HIV and AIDS. Now, this is a virus that is worthy to know about because it's something that is causing a lot of problems in our society today. It is a sexually transmitted disease. Now, we'll just learn, I'll give you a brief introduction, after which we'll go into looking at the structure of what the virus looks like, and then we'll now make mention of the replication and the normal life, the life cycle of this virus, and then how it affects man. Then we'll now learn about the natural cause in a human being. We'll now look at also the diagnosis and, of course, the treatment for this disease. Now, basically, HIV is just the full meaning is human immunodeficiency virus. And from the name human immunodeficiency. Now, this virus is targeted at the immune system of a human, and the aim is to suppress that immune system. And once the immune system is suppressed, once someone is infected by this virus over a long course, the effect on the human body will be that your immune system will now be depressed, or your immune system will become deficient. Though the exact immune system that is affected by this virus is the adaptive immune system. So that's adaptive immunity. So this virus will target T lymphocytes. So once they target those cells, not just T lymphocytes, any cell that has the CD4 molecule, like T lymphocyte. Other people we have is also macrophages. And aside from macrophages, we still have these other guys we call dendritic cells. So all of them that have these CD4 cells, those are my targets. Now, once I target them, I destroy them. I am depressing immunity, so that it is not even HIV that ends up killing the patient on the long run. These other infections we call opportunistic infections, which is one of the things we talk about today. But realize that as immunity is now depressed, the person is more exposed to normal diseases that would have not affected him. And those diseases are now what will ultimately cause the patient, in this case, to die. All right, so that just covers a brief introduction about HIV, but we go straight into the structure. HIV is a retrovirus, a retrovirus. So we commonly hear retroviral. The idea of retroviral or calling it a retrovirus is because of what it does. So it is a virus, and it is actually an RNA virus. Of course, we can have two types of viruses, either DNA viruses or RNA viruses. But for HIV, it is an RNA virus. That means it has its genome is an RNA strand. And of course, in the structure, we're going to look at that. So like every other regular cell, a cell should have a membrane. It should have what? A nucleus. A cell should have a cytoplasm. So that's a similar thing I would show you in the structure of HIV today. So in this structure of HIV I have here, I'll move up close. You realize it's like any other regular cell. This is the membrane, the membrane of the HIV. This would have been the nucleus if you would have called it so. This would have been the cytoplasm. But here they are given different names. This is still the membrane of the HIV virus. Right here we call this the capsid. The capsid. It is actually, some people say it is cone shaped. So it is the capsid we have in the center, and this other part here is called the matrix. So we'll look at the content of each of these parts, and then that's basically it for the structure. So when I look at the matrix, the capsid, the content of the capsid primarily is two single-stranded RNA, RNA guys. So we have two single-stranded. There are single-stranded. There are two of them. So that is my guy. Beyond the two single-stranded RNA guys, I also have enzymes. There are three in number. If you can see it, this should be. I just labeled them. The people we have will be RIP. R here is reverse transcriptase. I is integrase. And then the P is protease. So you observe that three enzymes. So there are two single-stranded RNA. Two single-stranded RNA genome here. Then there are three enzymes. Also, within the capsid, we have proteins. The proteins that are notable of concern here is the capsid protein, which is designated by P24, and then the nucleocapsid protein, which is designated by P7/P9. Now, the genome, that's the RNA strand, has three genes, and those three genes are the GAG gene, the pole gene, and if you can see this last one here, the envelope gene. So those are the three genes we have. But of course, what are we taking out from the nucleus, or would I say the capsid of an HIV virus, it is cone shaped. Yeah, I could say it's cone shaped. It has the genome here, which is our single stranded RNA genome. Beyond that, we have three enzymes: RIP, reverse transcriptase, integrase, and protease. We'll talk about what they do as we go on. And then also another thing we see that is part of this is now that it has proteins. The proteins here are either the nucleocapsid protein called P7P9 or the other protein called capsid protein, which is P24. Now, beyond that, the matrix has a protein, and that's matrix protein, and that's P17. Then at the envelope here, we have a transmembrane protein that's called G41. And then we have this other protein we call the surface glycoprotein, and that is called GP120. So, all right, so this surface glycoprotein, of course, as we're going to talk about it later, the GP120, the GP120 and the GP41 are formed within the human cell. So that's where their source is. So the Golgi apparatus just destroy, they pick up a particular guy we call, that guy has GP160 and we divide it to have GP41 and GP120. Yeah, I know there's an extra one, but that's what we are told. That's what the books tell us. Now, beyond this, other things that are important about the structure there. So the notable things is the capsid. What are the contents of the capsid? You got it right. You mentioned the contents. I've mentioned my DNA, my RNA, please get me right. RNA, enzymes, the proteins, and even the genes, the proteins and the genes. Then you go to the matrix, just the matrix protein P17. And then you go to the outside, the envelope. So on the membrane, we have the surface glycoprotein, which is GP120, and then we have the transmembrane protein, which is GP41. Something that is worthy of note. GP120 is what we use to bind to the CD4 plus molecules, whereas GP41 is what we use to bind to the co-receptor, which is either CCR5 or CXCR4. CCR5 is just the idea there is just it is a chemokine receptor. So CCR5, or we could have CXCR4. So that is that for the structure. Now the replication. So how is HIV transmitted? Basically, HIV is transmitted using body fluids, so blood and other body fluids. Now, notably, the main transmission is during sexual intercourse. So that's the main route of transmission. During sexual intercourse, when there is exchange of fluids between the partners partners that are doing the sex, during that intercourse, there could be transmission. That's the primary way, usually accounting for as high as 80% of the transmission of HIV. But beyond that, we have another one we have is a case where it's using of maybe blood transfusion, but there are a lot of tests currently done. Transition is not done without the right test. So we don't just pick blood from people. So for that reason, it reduces the risk or the occurrence of having HIV from blood transfusion, even SHAPs. So those are the other kind of horizontal transmission we have. Then we now have a vertical transmission, which is from mother to child. Please, it is important I mention this because vertical means mother generation to another generation, so mother to child. But when we say horizontal, that means in the same generation, which is either sexual or SHAPs and any other thing, maybe IV drop use and all of those things. So those are the ways we transmit HIV. Now, once a particular individual contracts the virus comes in and its target is the CD4 molecule on different cells. The notable cells that have CD4 molecules is number 1, T lymphocytes, T helper cells specifically. Number 2, macrophages. Then number 3 is our guy, which we call dendritic cell, like I said earlier. So my main note here, the main person we usually attack is CD4 molecule on T lymphocytes. So once they are attacked, the HIV virus would use its surface glycoprotein, which is GP120, to bind to this molecule. And then we now use I'll use the 120 and I bind to the molecule, the CD4 molecule. I now use my transmembrane protein, which is GP41, to bind to the co-receptor, which is either CCR5, notably, or could also have CXCR4. So those That's so once they bind, that it is now absorbed. So there will be fusion of the membrane, and once fusion occurs, the guy is carried in. That's the nucleic capsid will release its content into the cell. That's the T lymphocyte. So once they come in, the enzymes come in. We start with R. The function of R, reverse transcriptase. So a normal, what's transcription? Transcription is moving from DNA to mRNA. That's the normal way we know transcription to be. But right here, I'm trying to move from RNA back to DNA. So we call it reverse transcriptase. So it's a reversal. So they go backward. Now, so in this reverse transcription, my DNA is changing, my my RNA, genomic RNA, is changing back to a DNA, the enzyme, reverse transcriptase. And we are forming back a DNA, which should be how many? Double-stranded. So I'll just form back the DNA. Now, though we are told polymerase helps with an action, but I don't want to go into all of that. So once I form my DNA, that DNA that is formed from the reverse transcription of the RNA is called proviral DNA. So that proviral DNA is now carried into the nucleus of the human cell. This is the T lymphocyte or any other CD4 cell. I carry it into that cell, and within that cell, what I now do to that cell is that I carry it into the nucleus of the cell. Please understand something. After binding, there is endocytosis. So once I have my surface glycoprotein bind and the co-receptor bind, that is the GP41 binds to the co-receptor, they come inside. It is endocytosis. On coming in, my guy is transcribed. The RNA is now transcribed into what? Proviral DNA. Reverse transcribed into proviral DNA. That proviral DNA is now carried to the nucleus of that cell and integrated by the enzyme integrase. You see how these enzymes are adding up. So now, once they're integrated into the host genome, so that means I will put it into your own genome. I will now have to wait. That's the first phase of HIV replication. So everything I said up until integration into DNA, the DNA of the host cell, that is the first phase of HIV replication. Now the second phase now depends on the person's cell. So when your cell wants to replicate, it's normal replication. The HIV virus will now use the replicative power, that is the genetic machinery of your own host cell. And what does it use it for? Once I use your own genetic material, I pick your genetic material and then I replicate. So when you are replicating, me, I'm also doing my own. So now what it actually does here is that every cell should do transcription. So it will also do transcription. But its transcription will be forming two things. One will be a genomic RNA. And the other one will be it will transcribe to form mRNA. What's the difference? Genomic RNA is what I'll have that will now be found in the virus when I'm done with everything. So the genomic RNA will be this one here. That will be my genomic RNA. But my mRNA will now be used to form every other thing, the proteins, the enzymes, whatsoever else you need to have here. It is my mRNA that will help me form that. So once I come here, transcription to form genomic RNA and mRNA, then I bring these people outside. The mRNA now undergoes translation, and that occurs on rough endoplasmic reticulum, specifically on the ribosomes of the rough endoplasmic reticulum. After that happens, there's post-translational modification, and then of course I have these guys form, proteins form. Now these guys are now reassembled. So I'll carry the genome, I'll carry all these proteins and reassemble them. Sometimes the proteins are quite long, so we need them to be cut. So what helps to cut them into the functional proteins is protease. So you observe how these enzymes come in. So protease will help you to cut. And then, oh, you have cut the guy now. Okay, so we are good. So that is just that. Now, as I progress from here, they are reassembled. And after being reassembled, aside from that, remember I said something that GP120 and GP41 are produced by the host cell. So they are not even part of what these people are doing. So the host cell, its Golgi apparatus will work on. As the host cell will start working, and the endoplasmic reticulum of the host cell and the Golgi apparatus, they will work. And the end product of what they are doing is that they will produce GP120 and GP41 from a particular protein we call GP160. So when I produce these two people, I will now keep them on the surface of the host cell. So these guys only acquire that thing when they are going out. So when they want to bud out, they want to step out of the cell, like exocytosis. As they want to go out, they pick up those surface proteins, and they pick up the surface protein, and we have a new virus form, and that virus is called viron. Viron. So I repeat myself. This replication is simple. There is binding at the start, which involves your the HIV surface glycoprotein, and then the HIV GP41. Two of them will be binding to the receptor CD4 molecule and the co-receptor CCR5 respectively. After binding, there is entry, so they are brought in. Once they are brought in, there is now reverse transcription. The proviral DNA I'm forming will now be carried into the nucleus and then integrated by integrase. Then it uses the cell's machinery, genetic machinery, to replicate and then give us a new. It would undergo transcription, and what is it forming? A viral genome or RNA genome, and also an mRNA. So what do we have here? The mRNA will undergo translation to form protein and all of the things that we need in the virus. Then the genome will join together. They now assemble and they board out. As they are going out, they pick up the GP41 and GP120 as they are going out, and we have a new cell form called the viron. So they just keep going out, attacking many other people, but in the course of boarding out, they destroy the whole cell. So gradually, the whole cell is being destroyed. So that is just that. Now, what's the natural progression of this disease? It has 3 stages. The disease has 3 stages. The first stage is called our acute asymptomatic stage. So during this stage, it is acute. But so acute means it just happens quickly, and then at that moment, it is asymptomatic, no symptom. The patient could just have the regular prodromic symptoms. Prodromic means the regular symptoms people have, like malaise. Malaise is that you just feel unwell. You could have fever. You could just have those regular things that just signifies that there might be a problem, and one would just think maybe malaria or any other thing. So that's the first thing that would see, and that is in stage 1. Now, this stage, there is no antibody production yet, because these guys, the virus is replicating actively at this moment. But the only thing is that these viruses that come in, the body has not generated antibody yet to fight the virus. So if we should do a test at this period, there is no, listen very carefully, if we do a test at this period, it will not be positive. So that's why most times, if people get in contact, maybe rep or whatever case that they get in contact, and we suspect HIV, we ensure that they do a test, but they come back in 3 months to do another test. Why? Because there's a chance that if you just do the test immediately after the rep case or after the contact, you would most likely not pick HIV virus. So it has to, because the antibodies would not be generated yet against the virus. So basically what we do at this stage, we call it acute asymptomatic. We also call it seronegative, because we don't have anything in the serum. There's no antibody against the HIV virus yet in the serum. So that is that for first one. It just lasts for a short time. Some people say 3 to 6 weeks. Other people say it could take up to 6 months. But whatever the case is, that's the duration for the first stage. The second stage is called chronic, but still asymptomatic. Now, the symptoms are not quite, the person will still be having those feelings of unwellness. It's just that it's not so much. But the key thing about this second stage is that it is now seropositive. So if we should do a blood test for HIV, it will reflect positive, because the patient indeed is positive. So he has picked something. So that's seropositive. So that's the idea here, that it could be seropositive. Then what else about, and this stage, please, the viral load is increasing a lot. So the virus is replicating more and more. But then the lymphocytes are dropping. Why? They are dying off. So those are the key things here. If I do a blood test, I will see HIV. Usually we say it's around 6 months. So as high as maybe 5 years. So up to 5 years, the person could still be in chronic phase, which is stage 2. But after 5 years, people can now move into stage 3, which here we call, this is now the symptomatic phase. And please, stage 2 is called latent phase. So stage 3 is now called symptomatic phase, where we can have this disease actually happening. And beyond that, also another thing notable about stage 2 of this virus is that besides being symptomatic, stage 2 being symptomatic, stage 3 stage 3 being symptomatic, you can also have opening to the lymphocyte level is dropping so low that there is now opportunistic infection, of which the number one cancer that can affect an HIV patient is Kaposi sarcoma. Kaposi sarcoma. I'll spell the word Koposi. Sarcoma is spelled Sarcoma. So the skin, it's an effect on the skin. So we see this kind of rashes on the skin. So that's what we call Kaposi sarcoma seen in HIV patients. Now, beyond that, we have TB. TB is quite common in HIV patients. But in the hospital, when we are exposed to patients and we see that, oh, these people have TB, we try to test for HIV because most likely around Nigeria here or around the world, HIV is one of the key things that will depress your immunity and then predispose you to tuberculosis. And we have some other ones like the first one, pneumocystis, pneumonia. All of those things are just this. So this is a stage, those things will happen. And once your lymphocytes counts decreases below 200 international units per liter, at that very moment, we call it AIDS. So please take note of that value. Once you go below 200 international units, so lymphocyte counts below 200 international units per liter, is now called AIDS. So AIDS is just a severe endpoint of HIV, and that is just that for that. So now we go straight into diagnosis. In the diagnosis of HIV, basically we have just three tests we use for HIV test. The first one is usually called ELISA. ELISA, ELISA, full meaning is enzyme linked immuno assay, immunosorbent assay. So now usually we use it to just check the level of antibodies we have, that is against HIV virus, against HIV surface protein. And like I said, if you do this test early, after the person was exposed, most likely the person would be negative. But later on, it will now turn to positive. And once this test is positive, well, there is a particular test we use for confirmation. And that confirmatory test is called Western blot. Western blot. So please, the other one is just a screening test. What's a screening test? I check to see if you have the disease. But after I check and then I find out that it is positive, I use another test to confirm, because HIV is quite stigmatizing. You cannot just tell a patient you have HIV. What's that? So you need to ensure that, oh, you are sure. So the assurance test is Western blot, which is confirmatory. Now, beyond that, we also could use PCR, polymerase chain reaction, to also do something. Now, so that is just that for the diagnosis. And then we now go into prevention. Of course, the basic prevention is based on how it's transmitted. Practice safe sex, and beyond safe sex, that is sexual intercourse. Then also control the use of sharps. So you should have your sharps. You should have your the regular things, your clipper. Just control what you are using. It should not be used anyhow. So that is just that for that covers the prevention. Aside from that, mother to child, so we screen and we know. And then it could also be transmitted even in breast milk, so we avoid an HIV mother from breastfeeding the child. So what we do in that case is, when you have an HIV mother, we just move on to complementary breastfeeding. We use something else instead of, so we use breast milk, like we use milk, complementary milk. So yeah, that's that for base model I'm talking about. So now we go on. So other prevention, we have this particular therapy we call PrEP. So PrEP is just to help prevent you from even having prophylaxis. So we call it prophylactic therapy. It's just to help you prevent you from having HIV. And if we are told that if some doctors have to probably operate and they know this is an HIV patient, despite having to wear gloves and all of those protective, some of them could take PrEP so that they ensure they don't come down with HIV. So it's just a preventive treatment. Now, when we now go to the actual treatment, we don't really have treatment. The treatments are targeted at just stopping the replication. So the treatment measures are just, they're not to treat HIV per se, they're to stop the HIV from causing more problems. So if I discover HIV on time, I can start the treatment to now ensure the person stays alive. So I suppress the viral load and then I prevent more destruction of my lymphocytes. So that is just by targeting some key places. One of which, quite common, is that we target the reverse transcription. So those guys are usually called nucleotide. And we have these nucleoside people that can target reverse transcriptase. We also have people that are non-nucleoside reverse transcriptase inhibitors. So we just give the name nucleoside N, reverse R, transcriptase T, inhibitors I. So those are NRITs. So those ones will inhibit transcription. So they are only just, they come in as a nucleoside, but they block the elongation. We have these other people that just come in, they don't even join, they just block. We have people that could prevent you from even binding. And beyond that, at different points, we have people that could inhibit the protease. We have people that could inhibit the integrase. But at different points, these guys are inhibited. So yeah, that is just that about HIV. I believe you understood. If you have any question, please drop it in the comment section. We are going to be hosting a Q&A session on this, on HIV, which will clarify every form of issue you could have with it. And then beyond that, this is Effortless Mastery. Do well to like, do well to subscribe to this channel, do well to share this video across to as many people as possible. This is something that can help you, can help many other people, and then can surely help any medical student that has to prepare for an exam. So thank you very much for joining us. Until next time, God bless you.